Ahead and get started. I'm going to introduce our lovely speaker today. Noel Jee was previously a principal at Illumina Ventures with a focus on therapeutics and diagnostics for the last 4 years. Prior to joining the fund, Noel worked at LEK Consulting as a management consultant specializing in the life sciences. He has consulted on strategy engagements for companies in the pharmaceuticals, biotech, and diagnostics industries. He's also obtained a dual BS degree from University of Maryland, College Park, and his PhD in chemistry and chemical biology from the University of California, San Francisco. My good friend Rajiv also says he's the smartest person he's ever met, and I tend to agree with him that Noel is crazy smart. So with that, Noel, why don't you take it away and jump right into the first sort of section of your presentation.
Making sure I'm off mute here. Thanks for the nice intro, Alex. Always too kind. I told Alex to tone it down a little bit and set expectations a little lower this time, but as I'll try to meet them. It's a pleasure to teach this seminar series on statistical literacy. A few people asked, why am I teaching this? Well, Alex asked me to do so, and there's a few things I'm going to hit today, talking about a few different frameworks in how I think about statistics when it comes to technical diligence. But as Alex mentioned, please make sure that you submit questions or ask questions, because we will probably only cover half of the stuff that people actually care about, and then the other half, I'm assuming, we'll do in the Q&A. So to start, the first seminar is called Think in Distributions, and I'll go into what this actually means. Just quick background, I'm not gonna belabor this. I have a chemical biology PhD, spent 4 years at Illumina Ventures, tried to do startups twice, didn't work out, but that's okay. And then a few disclaimers like, these are not investment advice and everything's my own view, etc. So I'm gonna start by talking about a company called Banana Bio. Obviously, I made this up, but it's kind of a company that I've made up based on a lot of real-world experience. And I'll use this as an example throughout this entire first seminar. So the company is called Banana Bio, and it was the hottest company making the rounds, great team from top universities. And you see, when they were in academia, they had independently discovered this new biological phenomenon, something called bananas. And it turns out that cells use bananas to send messages to each other across the entire body. And each banana is like a chunk of code, and cells eat them to decode the messages within. I kind of took this from real life, and this was actually a Nobel Prize winning discovery. But the company found a way to capture these bananas, decode them in the lab, and then use them to diagnose diseases. And what they found was astonishing. And some of the data that they provided from these discoveries are shown here. So it turns out that these bananas could be used to detect many types of cancers, and they could even detect complicated diseases like lupus. And the company was able to make diagnostic tests with near perfect AUCs across all of these different diseases. But as the title implies, amazing numbers can be deceiving. And so in this session, I'm going to use this company as an example to walk through some fundamental statistical concepts. For example, to understand what an AUC is, we must first understand what a ROC curve is. And for that, we need to understand how sensitivity and specificity are calculated. All right, so this is going to be fun. Let's start with some very, very basic things here. So to really understand statistical concepts, we have to start with the framework, or people like me with average intelligence have to start with the framework, because I don't know everything. And my framework that I'll describe here is thinking in distributions. So just to be thorough, let me quickly walk you through the basics. This is probably review, third grade review for most of you. This is a normal distribution, and you can have a right skew or a left skew. This is a right skew that I drew out. I'm sorry, this is a left skew that I drew out. It's not perfect. It looks kind of like a normal distribution. And this is a right skew. I just imagine, pinching the side of a normal distribution and dragging it out. And so where the tail is, if the tail is to the right, then that's a right skew, and vice versa for the left. So, you know, in a normal distribution, you have the mean, median, mode all centered. But when you have a skew, you basically pull the mean in one direction. So with the left skew, you're pulling the mean to the left, and alongside of it, the median also gets pulled to the left, and the mode kind of stays where the hump is, right? Because it's where the highest number of the same variables are. There's a lot of different types of distributions, and some of them are mentioned here, but I'm going to skip over talking about lots of different types of distributions because you can just look them up online. One thing to quickly note is that a lot of times companies and people in research talk about Poisson distributions as Gaussian distributions. They're basically very similar. It's just that Poisson distributions use discrete numbers, so like whole numbers, and Gaussian can be contiguous. So that's the difference between those two terms. One thing to remember is that while we always think of a smooth curve, distributions are fit to messy data sets. And so the smaller the sample size, the less perfect a distribution fits the data, right? And so if you only have a few kind of variables, then your distribution curve looks great, but you can see that it's quite messy. And so this is a framework, and it's really useful when thinking about statistical concepts. So you should understand the point of this is to know when to apply this. A lot of times I use it as a baseline framework, but it's very useful when thinking about things like sensitivity and specificity. And I'd argue that this is probably the easiest, most intuitive way to think about sensitivity and specificity. So there's a question that came in a while ago about, why does sensitivity and specificity always have to trade off? Well, let's think and apply this framework here. Sensitivity and specificity should be thought of as distributions of healthy and diseased individuals. So this is a healthy curve. Everyone under this curve is healthy. Everyone under this disease curve is diseased. Now, in more scientific terms, usually when you have a disease curve, this blue curve here, you call them, true positives. And for the healthy, you call them true negatives because they're negative for the disease. Since these distributions overlap, we need to set a threshold shown by this line here to call everyone to the left of the line healthy and everyone to the right of the line diseased. That makes sense, which makes these parts of the distribution misclassified as false negatives and false positives. And if you take this threshold line and just look at the blue distribution, the sensitivity here is defined by everything that's under the blue curve to the right of this threshold line divided by the entire area of the blue curve. And specificity is the same thing except for the green curve. You're taking the true negatives and you're dividing the true negatives by the entire population under this healthy curve. And so that's why there's always a trade-off between sensitivity and specificity. If you shift this line, this threshold line to the left, then you increase sensitivity, right? Because now you have more true positives that you would call. But when you move the threshold, you necessarily decrease the specificity because now the percent of the healthy, the true negatives, divided by the total number of healthy people is a much smaller percentage. So your specificity necessarily decreases. That's why whenever you see distributions like this between healthy and disease, you always draw this threshold in the middle where it's a nice kind of balance between sensitivity and specificity, but you can always tweak the threshold to increase sensitivity or specificity depending on what you need in the real world. I think people might ask, why not just make the curves separate a little bit? And that's exactly what technologies do, right? So different technologies change distributions to decrease these trade-offs. And when I say technologies, they can mean a lot of things like DNA, RNA, bananas, fancy algorithms, etc. And that's what changes the distributions to decrease the level of trade-offs. And that's actually how you get like perfect accuracies. but more on that at the end. Okay, so we understand sensitivity and specificity. So let's move on to the ROC curve. So ROC, ROC, stands for relative operating characteristics. You don't ever have to think about that again. Everyone just calls it a ROC curve. And the concept is simple. You're basically graphing out all possible sensitivities and specificities in this region here. So, if you want a perfect specificity, you move the threshold line to the right so that the green curve, all of it, is to the left of this threshold line. So you will not have any more false negatives, right? So if the way that you graph out a ROC curve is you literally take this threshold and move it all the way to the left. And at each point along here, you have a different sensitivity and specificity. And then you just basically map that out on a chart. And that's exactly what these lines are. So on the y-axis is sensitivity. On the x-axis, it's specificity, but it's 1 minus specificity, but it measures the same thing, so the false positive rate. So if you have perfect sensitivity and specificity, you get this green line called the perfect classifier. And if your distributions overlap, we have this red line, and everyone else is in between. And you can imagine what AUC means. It means area under the curve. And you want to move as close to the green line as possible to get you an AUC that's a perfect 1. Right? That means there's absolutely no overlap between these two distributions, and this yellow area goes to 0. And so you can imagine what a ROC curve looked like for Banana Bio when they gave these numbers. If the AUC is 0.97 or 0.94, there's very little wiggle room for what the ROC curve can look like. It's going to be as close to the perfect classifier as possible. But you have to also be careful when the company talks about AUCs, because sometimes the shape of the curve matters. Because most companies do not have AUCs that are in this 97% range. All right, so there's different applications where sensitivity is more important than specificity and vice versa. And we can chat about this later. Sometimes you're talking about screening versus diagnoses, and you have different trade-offs for different types of tests there. But the distributions look different, right? When you look at the shape of the ROC curve, it tells you about which is higher and which is lower. So if the distributions are different, sometimes you might have higher sensitivity versus specificity. The AUC itself does not tell you what the differences are between the sensitivity and specificity. So it can be quite deceptive, and you have to look at the shape of the ROC curve itself to see whether or not things are pretty much down the middle, or if it skews more towards sensitivity, or if it skews more towards specificity. I can say that in the case of a company like Banana Bio, where they're giving you an AUC that's like 0.97, it's hard to imagine that there's any room for shenanigans. So that's all great, but everyone always shows you good data. So how do you know if something's too good to be true? So I took this one example from the slide where Banana Bio shared all their amazing AUCs. These AUCs were nearly perfect, which means there's probably not a whole lot to trade off, right? And if you look at the actual ROC curve, it's nearly perfect. I mean, I just made this up and I drew it, but this is probably what it looked like. But obviously I wouldn't use this example if there wasn't some twist. If you unpack what the company told us, we know that they're not just using the banana factor, they're also using this thing, RNA, to get to this ROC curve. And so they're measuring other known hallmarks of disease. And that makes perfect sense. Why not use everything at your disposal to make the best test possible? That's what I would do. But buried in some background data, not in the pitch deck, and not even in their supplemental information, but buried in one of the paper's supplemental information, was this pretty chart. And it turns out that RNA alone gave you a pretty good AUC of 0.94, and the banana factor alone was pretty terrible at 0.64. And so you combine these two to get to 0.97. That means the banana factor really only increased the AUC by 3%, an absolute 3%. So the thing is that you have to deconstruct the data to find what's hidden, and deconstructing this data is necessary to find what's hidden, but it's not sufficient. As an investor at this point, I would look for context as well, to figure out if, just because this banana factor adds very little, is that still something that's unique and useful? Because sometimes a small difference can mean a lot to the patient. So maybe this 0.3% is actually something that doctors care about, and maybe it changes how they care for patients. They don't, and it doesn't. But if it was important, deconstructing can lead you to ask these other questions, like why is RNA line so good? And is this real in itself, right? What does the literature say? What do other labs that have published on this have to say about this? What does a sample look like? This is a topic of the next session, by the way. And I kind of took this from a real company. We did not actually invest in Banana Bio at Illumina Ventures. All right, I'm going to move beyond sensitivity and specificity and talk about kind of the bigger picture, and in which you have to also consider this thing called positive predictive value and negative predictive value. This is something that you might be familiar with, just given that we are at the tail end, hopefully, knock on wood, of a global pandemic in which testing was a very large part of how we handled the pandemic itself. So this is a standard chart to calculate everything. I'm not going to dwell on it or explain it. You can screenshot, or I think the slides are out there. You can look at this later. But sensitivity is a percentage of the true positives in the distribution of all diseased people, as you can see here, and specificity is the percentage of true negatives in all healthy people, by giving you an example. And let's talk about COVID testing because it's been so relevant. So a company might present this data showing the results of tests that they collected for 1 million people. So remember, sensitivity and specificity are down here. You're dividing the number of true positives by everything, right? So you end up with a 93.8% sensitivity and a 95.6% specificity. Looks pretty good, right? Well, yes and no. So sensitivity and specificity are pretty good, but the positive predictive value, which is calculated horizontally, is the probability that for somebody who tests positive, are you actually positive or not? And it's the same for the negative predictive value. And if you calculate that out, the positive predictive value is only 53%. So this just means that if you have a positive test, there's only a 53% chance that you actually have COVID. So this is really important because the positive predictive value is dependent on the actual prevalence of the disease. And so, you probably want to know why this number is so low, even if the sensitivity and specificity seem so high. It's because there's only 50,000 diseased patients, right? The total number of diseased patients is 50,000 versus 950,000 healthy patients. So million tested, 50,000 is just 5% prevalence. So that means that even with a greater specificity, you'll have a large number of patients in the false positive box, right? So specificity seems high, but when you're talking about 5% of 950,000 people, you end up with a pretty high number. And because a positive predictive value is calculated off of the true positives divided by all of the people that tested positive, and this non-disease number ends up being quite high just because it's a 5% prevalence of a very, very large number, your positive predictive value ends up being quite low. It's basically a coin toss. So you can test positive, but it's a coin toss whether or not you actually have COVID. So that's been a real problem with COVID tests, and part of the reason why a lot of people had a false sense of security. But yeah, I mean, if you are told to get tested, you should still get tested. But it's been a challenge. It's been a challenge for the CDC. It's been a challenge for academics to figure out what to recommend in terms of testing. I'd actually say that in today's world, it seems like people have gone from saying, take a test that has high sensitivity and specificity versus just take a test that's cheaper and faster and that has much lower sensitivity and specificity, because a positive predictive value ends up being about the same. So being tested more frequently, with a more convenient test ends up being better for the population than a highly sensitive or specific test. This also should translate to companies that are selling diagnostic tests. Just because you have a high sensitivity or specificity doesn't mean that you have what the market actually needs. So another thing for investors that are listening in, don't be fooled into companies when they pitch optimal numbers. It's not just about the highest numbers. It's about what the actual market requirements are. And sometimes it's a lower sensitivity and specificity. So these great numbers can still lead to a useless test. And this is hyperbole here, but it's important to understand the whole picture. And I'll use one more example to illustrate this. So I just talked about in COVID testing, how people have been, experts have now been recommending taking a more convenient but less sensitive and specific test. And so there's 3 different types of COVID tests that are available today. One is a PCR test, which is looking for the RNA of the SARS-CoV-2 virus. There's an antigen test in which you're looking for actual protein components, so parts of this virus itself. And then there's antibody tests, which are looking for antibodies that you produced as a human against the virus itself. And there's different trade-offs in all of these. So I'm not going to go over the components of the virus. You can look that up yourself. But the most common test out there right now is a PCR test. And this looks for the RNA in the SARS-CoV-2 virus, as I mentioned, and it creates an amplified DNA library that's detected downstream. And sensitivity specificity is typically above 90% for these tests. The antigen tests, which are becoming more and more available, I think you can go to Walmart and buy some of these tests for at-home use now. Actually, you can do the same for the PCR test. They look for protein components of the virus itself, but you can't amplify protein components, right? You can amplify DNA, but you can't make more virus from a pre-existing virus that's dead and fragmented. And so that's why the sensitivity and specificity tend to be a little bit low for these tests. I'm not really going to talk about antibody tests, although if people are interested, there is a clinical trial that's free for people right now that you can take that looks for antibodies to see if you've ever had a reaction to COVID. So if you feel like you had COVID, but you were never quite sure, you should sign up for this study. They'll send you a little kit to draw blood, and they'll tell you if you ever had COVID. It's also fun. You can also go back once you had your vaccine, and then they'll tell you if you have antibodies against the synthetic COVID that was injected. All right, getting back to the COVID story here. So there was a company that developed an antigen test, and they claimed 96% sensitivity, which sounded amazing, but real world testing found that it was more like 80%. So this is where kind of the independent analyses come into fruition. So the company claims 96%. Actual clinicians at the Mayo Clinic published a paper saying that it was only 80% in the real world. So when you have a clinical trial, remember this as well, things will always look better because it's such a controlled setting, versus when you have things in the real world, just take 15 or 20% off the top of whatever people say the efficacy is for a diagnostic or a drug or something else. So even with an 80% sensitivity, the trade-offs were worth it because the turnaround time was like 15 to 30 minutes. It was less than half the cost of a PCR test. I think it was like 20 bucks when PCR was costing 120 bucks. So it sounded great. It sounded like exactly what the market needed. But if you look at, let's unpack this. What does the 80% actually mean? How was the sensitivity number derived? Well, in this case, 80% of the patients, 80% sensitivity means that 80% of the patients were tested positive with a PCR test. So another point here about unpacking the data, figure out what true positive actually means, because it's a relative phrase. How do you know in your curve who is healthy and who is diseased? You have to have someone say that these people are healthy and these people are diseased, right? And so in this case, the baseline for what a true positive was is a PCR test, and a PCR test is not positive or not 100% accurate, right? So you have to know that the sensitivity already has a margin of error built into it because it was comparisons. It was done with comparisons to a PCR. Well, a funny thing that happened here is that this is a real company. It turns out that the sample they used were symptomatic patients with COVID-19. And what the experts were recommending is that you test everybody frequently because the test is so cheap, and even with a reduced sensitivity, you can just do it frequently on all these different people, and it should help stop the spread of COVID. This was many months ago. But it turns out that if you include asymptomatic patients, the sensitivity was reduced below 40%. So this also goes into remembering that sample itself is very important. We'll talk about this in a later lecture. But depending on the market needs, if the market needs a test for everyone, because experts are saying test everybody, including asymptomatic patients, you have to understand as an investor, what that actually means for the product and the sensitivity, specificity, etc. In this case, the company's sensitivity fell below 40%, including the asymptomatic patients. There was a big blow up, and this is a big publicly traded company, so you could look it up. I think the New York Times did an expose on it many months ago. All right, last thing, just kind of for fun, is that sometimes numbers mean nothing. So there's this term accuracy that I've been throwing around. So accuracy in the context of diagnostics, at the very least, is defined as true positives plus true negatives divided by all the tests that were given. So 90% accuracy seems great, but it could actually mean nothing depending on what the test is actually used for. So in this case, it could mean that you have 90% of your true positives that were captured and 0% of your true negatives that were captured, and you still have a 90% accuracy, but your specificity is 0. And specificity actually does matter. So when a company throws around terms like accuracy, remember that it doesn't actually mean anything. You have to dig deeper into what the actual sensitivity, specificity numbers are. And even then, you have to unpack that further into positive and negative predictive values. There's a fun thing that I like to say, sometimes in pitch decks you see this combined team experience of over 67 years. The hell does this even mean, right? Combined experience of 67 years come anywhere close to like a single person with 67 years of experience? I don't think so. I think it's an absolute garbage number. I always tell startups, if I tell them this number doesn't mean anything to me. I kind of laugh at it whenever I see it. They ask, what should we do instead? I usually say just tout why each member is relevant to solving a problem that is part of the startup. It doesn't really matter. We're in Silicon Valley. People invest in young companies, young startups, young founders. This type of shenanigans, I don't think flies anymore. I'm going to refer to this here. Sometimes numbers mean nothing, even in big data that is used to move stock prices very high. And I'll talk about this in the third seminar, but I'm teasing it here because I think it's really interesting. I'll come back to this data later. It's not just startups, it's also publicly traded companies that play around with data to show something to be potentially better than it actually is. All right, here's an obligatory kind of fun quote slide at the very end. I'm sure everyone's heard this quote before. Well, I dug into it. It turns out that this was actually a wrong quote. So I think Benjamin Disraeli was attributed to, or was quoted saying this and attributing it to Mark Twain, and it never actually was said by Mark Twain. All right, here's a break. Thank you, everyone.
Awesome.